COVID era. However, my name is Bola Obo. I work in the University of Lagos. I am the Director Research and Innovation Unit, but my department is the Department of Cell Biology and Genetics. And so I'm a professor of genetics. Having studied plant science, I've done a lot of biology as my first degree. My areas of work span plant genetics, animal genetics, population genetics, conservation genetics. And that is what I have done in the past about 30 years since coming over to the University of Lagos. My initial stint was in the Nigerian Institute for Oil Palm Research in Benin City. Yep. So relevance of population genetics in a post-COVID era. Relevance of population genetics in a post-COVID era. Well, I will look at it from the point of agriculture. I will look at it from the point of, are we relevant in health? And I will look at it from the point of social sciences and humanities. Population genetics, we all know, is a field of biology. Population genetics is a field of biology that studies the genetic composition of biological populations, the changes in the genetic composition that will result from various factors, including natural selection. And so in population genetics, we try to apply Mendelian laws and other genetic principles to the entire populations of organisms and not only individuals. We also try to study gene frequencies, and so it's closely related to evolutionary genetics, because we know that evolution at the end of the day depends heavily on changes in gene frequencies. So the key concepts in population genetics will be, we are looking at the population, we are looking at genotype frequencies, we are looking at the gene pool, and we are looking at the population itself. And so what has been happening in agriculture?
I'm sorry, Prof, to interrupt. Can you please share your screen if you have presentations?
I don't have a prepared screen, please. I can put my video to see my face, but I don't have.
Oh, okay. We'll try and follow then.
Okay. I could send the slides, prepare something and send to you later to share with members.
Okay, that will be fine. Thank you.
All right, that will be okay. So population genetics, our populations are exploding with urbanization. We have a decrease for those of us in Lagos and environs. We see that with industrialization, housing projects all over the place, cultivable lands are decreasing at a very fast rate. And this is leading to food insecurity. And so more than ever, we need to capture our plant genetic diversity. Before COVID, yes, we used to store all this in form of gene banks, DNA libraries, biorepositories, reserves, and all that. And that is how we have been storing our materials over the years. However, we need to do more to be able to have food and nutritional security. And so we need to provide tools as geneticists to be able to monitor the populations and to be able to assess what we have within these populations that will be useful for conservation planning. Because we need to conserve the more now. Also, when we begin to look at the impacts of climate change, we notice it is already noticeable that with the impact of climate change, there's a lot of decrease in variability of the genetic loci that controls both the physical and phenotypic response of an organism. And so the genetic variability and the ability of the population and species to persist despite these changes over time is becoming increasingly important. And so in order to conserve, we begin to think of how do we maintain optimum population size for conservation purposes, because it is what we conserve that would eventually be used in our crop improvement studies. So optimum population size is key going forward, because if we don't have the optimum size conserved, then that would already introduce a bias into our diversity assessment. And that will begin to have wrong predictions at the end of the day. We also know that some populations are becoming locally extinct. And this is due to the narrow range of genotypes and more phenotypically uniform accessions of varieties. And so we are beginning to have crop failure, materials fail to survive. Some survive and they fail to reproduce. So how then, in this post-COVID era, do we intend to be able to work harder in order to ameliorate the issues of food insecurity, which had been with us even before COVID? And so we need to have more comprehensive selections. And even now with climate change, we need to go on and begin to study whether you call them minor crops, you may call them orphan crops, you may call them underutilized crops. We need now to begin to study some of them because they may pose the repository for the future food baskets. And within the Nigerian context, I think we have a lot of them. Some of them are gradually gaining prominence. We have the Fonio group, the locally called Acha. We have the African yam bean. We have the pigeon peas. These are crops we may begin to look into in order to help our agriculture and food insecurity going forward. Yes, we know that once we conserve, we tend to assess diversity. And in the past, we've assessed diversity morphologically, doing a lot of morphological, quantitative, qualitative measurements most of the time. We also went into qualitative measurements. Another way in which we used to assess is the biochemical method using enzymes, allozyme studies, and all that. But most importantly, the use of molecular markers. And I think in agriculture now, that should actually be our main focus. We should begin to do a lot of work with the molecular markers. They are most widely used, and we know that they arise as a result of either DNA mutations, insertions, deletions, errors in replication, and all that. But we find that these markers are widely used. They are not limited in number. They are not affected by environmental factors. And so that is a plus for us with climate change. And the developmental stage of the plant does not also affect them. And so we can have a big data base, our molecular markers. And there are quite a number of ways in which all this can be analyzed. We can analyze them, studying their genetic relationships, analysis and samples, calculation of their genetic parameters, diversity, and the diversity can be partitioned at various levels. We measure a lot of distances, done a lot of things. My colleague will be talking on path analysis. Yes, we do that with our data. With that collected at the deep, we can begin to look at allelic genetics, similarities between two genotypes, similarities between populations. There are various statistical tools. A lot of softwares have been developed. We calculate the Ni and the least coefficients, distances, we analyze component dendrograms, and I think they help us at the end of the day to speed up the work of the plant breeder. And going forward, we may find that we may not be able to do extensive field work, like I said. However, with our collections and analysis at the molecular level, we will be able to pick upon the traits. Usually yield and nutrition is what people are looking for in all the crops now. We may be able to pick on that and help the breeder who is going into the field to quickly develop or introduce new varieties or new accessions. When we look at the area of health, availability of DNA sequences provides a lot of background. And I don't know whether there are any health practitioners in your midst, but in the area of epidemiology, we need to, COVID has taught us that there needs to be constant surveillance in order to determine population frequencies of the various genetic variants that can lead to specific diseases. Malaria is still endemic in a place like this, and there are lots of variants within the Anopheles and the Aedes species that cause malaria, that cause yellow fever. In our environment here in the University of Lagos, we find when we did our own surveillance and sampling years back, that we have a lot more of the Aedes species, which could also cause yellow fever. And there are communities like that. While we also find that there are quite a lot of variations in the Anopheles, and some of these variations, you find some of them resistant, and you find that is why there are a lot of malaria drug combinations now, because some are resistant to particular medications, some are resistant to even our common insecticides and all that. So there is need even in the health sector going forward to be able to marry population genetics with our surveillance studies and also see whether we can provide predictive models. Is it possible to have a predictive model that can help us predict emergence of a new disease? Is it possible to tell us the particular host size of a population is increasing and its density is increasing? And if it continues at that rate, a new disease may emerge that may be successful? Or is it possible to say, yes, the populations and their densities are gradually decreasing, and as such, we would not have successful disease outbreak in particular areas? So we really need at this stage to begin to marry the ecological aspects with the genetic aspects and be able to really track virus emergence and see whether we can have population genetic models that can help predict where and when the next zoonotic disease will start from. I think whether we like it or not, geneticists play an important role in this. Another thing that has crossed my mind, which I would like us as biometricians, population geneticists, to look at is where exactly can we be married with social sciences and humanities? Is there a relationship between us? Is there a relationship between us at this stage? So those are the things we would need to begin to consider going forward and see how we can help and how we can make an impact in this area. I think our ability to be able to predict is very important in the nation at this point in time. Also, our ability to be able to help predict, even when we go back to agriculture, what can we say about our yields going forward? What can we tell governments will be based on what has been cultivated this year, and if there's going to be an increase going forward, what would be the kind of yields we'll be expecting from our major crops and our minor crops? So I am looking at us really moving forward as biometricians, population genetics in our various fields, and actually bringing an impact, a practical impact in our environment. And yeah, so I think I've taken quite some time, and I hope I have teased our minds a bit on what we may need to be looking at and what we may need to do going forward. Thank you very much for the opportunity to be with you today.
Thank you very much for that presentation, Professor Obo. One thing before I hand over to Professor Omolara Olani, who will be sharing. Hello, Dr. Dauda, are you saying something? Okay, as I was saying, thank you, Professor Obo. One comment I want to make before I hand over to Professor Omolara Olani, who is supposed to chair this scientific session. Can you please share your video so that we can see your face at least once, so that when we meet on the streets, we can recognize you and give you a thumbs up at least. We just want to see your face.
And then, but mainly like that. I think you should think about it. Hello? All right.
Hello?
Sorry.
Yeah.
No, how can he not? I have told him now.
Hello? Let me invite Professor Mrs. Omolara Olani to chair the scientific session where we take comments on questions from members based on the presentation that was just made. Professor Olani, please.
Thank you very much, Honorable Secretary, Mrs. Olayemi Oluwashoga. We need to also thank Professor Obo for this session. Secretary General, do I have to put on my video?
You may.
Okay.
You may.
All right. Thank you very much.
Before you go on, let me say, if you have a question, can you please raise up your hand so that we will know and we can manage time as much as possible. I can see one hand up already, Dr. Shirali, so that the chair can use that as a guide. Thank you.
All right. Thank you very much. If we have only one person to ask questions, then it means that we will be able to save much time. Let me. Sorry, please. If we have only one person, it means that we'll be able to save much time. But let me quickly just give a brief of the presentation of Professor Obo. She had challenged us about
give me an ambulance. They are coming.
Okay, please, Dr. Gubadia Kenneth, can you please put off your mic? Can you mute your mic so that we don't have interferences? Thank you. So she had challenged us that we need to see how biometricians relate with different fields. She talked about genetics with special reference to agriculture and then to health. And she's asking relationship with social sciences. The question is, do we have data bank in Nigeria? Do we have what it takes to make these predictions? She has challenged us that we need to develop predictive models. How feasible would that be? If there is nothing close to what should be, what will be our plan towards, contributing to this nationally? With this, I'd like to recognize those who want to ask questions, Dr. Oshuolale and Dr. Yamaki. Can I give Dr. Oshuolale the floor, please, to ask his question?
Thank you very much. Yeah, let me first appreciate Professor Bola for that beautiful presentation. Well done, Prof. More power to your head, Buma. Yeah, I just want to briefly say one or two things about the virus, SARS-CoV-2. SARS-CoV-2 breaks down people as rapidly as possible, especially those with underlying illnesses. But we realize that some people will be tested positive, and later on they'll be tested negative, even without medication. Could genetic factor be responsible for this? That's my first question. The second one, as a geneticist, I've seen a case whereby I have an individual who has gone for test for genotype, and the test came out AA. After marriage, she was asked to go for another test, genotype. it came up AS. We asked her to go to another general hospital. It came up AS. What could be responsible for this change? Those are my two questions. Thank you, ma.
Can I take the question?
Yes,
now,
please, Professor Obo. You can take the question. We have only 2 people, so it's quicker.
Okay, thank you very much. For the first question or comment, do we have data bank for predictive models? I would say yes and no. Having a repository where we'll have all the data for the various things, no. But in our various establishments, and that is why we need this collaboration, in our various establishments, over the years, the work we have been doing, we have the data. And so having a society like this, it is good for us to collaborate further by bringing all that data together. And then as a society, we can begin to work on it. I know that if we talk about Greek, there is quite a lot of data in various establishments in the agricultural field. I know that in the medical field, it is a bit difficult. They are scattered in some places and in some teaching hospitals. But we need to work together to be able to begin to utilize what we as individuals have within us to be able to make a start. And that is why I said yes and no to that question. I believe we have to start from somewhere. We may not have the repository in a place as of now, but if we start from somewhere, both the government and the various establishments that are supposed to relate with us, actually even the, what do you call it, Office of Statistics or whatever, once we start from somewhere, we begin to relate with us the more, and then they will know the kind of data they even need to be collecting on a regular basis that would help us assist them to come out with most of these predictive models. Those are my thoughts on that. The second question says, could genetics favor SARS-CoV-2? Well, from the little I did here with some colleagues, there are a lot of factors that, genetic factors that helps people who come down with this COVID-19. And one of them is the blood group. Some studies have shown that the O genotype, usually because they are able to fight cardiovascular diseases better, don't have severe COVID-19 breakdowns, while those with the A are the ones that are really predisposed to having a lot of breakdowns, especially if they are comorbidities. In Africa, there's also the suggestion that could the BCG vaccine, which we know is compulsory here even till now, could it have helped? It had been stopped in Europe and most of those other countries 10, 20 years ago. Could it be the one giving us the kind of immunity we have in Africa in general? Another thing is, could our vitamin D levels, well, those are the things where we say we don't have data. In countries where there are data like India, they have said that their vitamin D levels have helped them to cope with COVID-19. So there are a lot of things that may have helped us in the African and Nigerian setting to cope with. But these are some of the suggestions in the various publications that have arisen in relation to COVID-19. For the genotype change, I will just say from my experience and what I have done that definitely that first test was a mistake. I would not believe that because of marriage, somebody's genotype will change from AA to AS. And then it's been done in 2 other places. And could there have been an error when her result was given to her before marriage? I think we should begin to interrogate such things and not begin to think that there was a change or there was a mutation. I'm not a hematologist, but from the little genetics I know, I don't see how between being a spinster or bachelor and being married would have caused a change in the genotype. Yeah, thank you.
Thank you. Thank you very much, Prof. Unless probably a miracle happened. All right, so let's invite Professor Oyamaki.
All right. Oh, sorry. Sorry, just let me. Okay, I believe you can hear me now.
You may put off your other device. Do you have your phone also connected?
No. Sorry, I'm a bit close to the control center. So
okay.
Okay. I guess it's better now.
Not so.
But I will just move away from there.
Okay. Or we take another question while you just sort that out.
Yeah, okay, I think we have to bear with Oyamaki. He's actually in the control room. He can't move further away from the systems here.
But I guess I can speak now.
Oh, now it's better.
Yeah, it's better.
Okay, I actually enjoyed Prof's presentation, and Prof has actually raised a lot of issues that I feel border on the mandate of IBS. She asked the question if there are a relationship between what we do and the expertise we could get from sociology. I think when we're talking about defective and defective data, some of these data lie in Africa. Most of the differences that we see in the new knowledge that is provided scientifically had some element of behavioral changes, behavioral sciences in them. And I think it is high time we started talking with people with sociological background and other areas of humanities, because there have been some areas or some expertise that actually impact science. And that will lead me to the second aspect, which borders on collaboration. I think most of the time we talk about this collaboration, but most of the time we don't we don't implement them. I wouldn't know how many teams that we have been able to develop now, even in IBS, that has, that has a multidisciplinary, multidisciplinary concepts. For example, most of the issues raised on predictive modeling. A good statistician may not be able to talk adequately to genetics if he has not been working with somebody with expertise from that has a good knowledge of that area. The same way, a good statistician may not be able to talk adequately to an agriculturist or an agronomist if they have not been working together. There are expertise in statistics that impact some of these areas, but because we don't talk together, because we don't have groups, because we don't have platforms, we have suffered in the aspect of having an adequate repository where we can actually solve most of the problems that we have. And the final one I want to talk about, which is a challenge to everyone, is that we should start developing platforms. Platforms whereby a geneticist with backgrounds in agriculture will talk to experts in statistics, experts in humanities, experts in every area that has to do, with some of the issues Because like I said, the way the culture of the people has a long way, in impacting the kind of science you do among them. So one way or the other, we should start speaking to ourselves. And I believe by the time we do this, we would have developed competencies, of a new generation of researchers that will be able to tackle some of these issues that we raise. Thank you. Thank you for your time.
Thank you very much. Prof, before I ask you to respond to that, there is a question in the chat that I got. I would like you to address it because it relates to genetics directly. That if mosquitoes are genetically evolving to resist drugs and insecticides, why are human beings not developing resistance to malaria and other mosquito parasite-borne diseases? So you may take that before you respond to the last question. Thank you.
Thank you very much, Professor Oyamaki. I think most of what he has said is actually food for thought for the society and the group, encouraging us for multi-institutional, collaboration, and going where the data is, and being able to show relevance, and then working with other groups. So I totally agree with what he has said, and I think that is one of the ways forward. Yes, mosquitoes developing resistance. Why are human beings not developing resistance? Well, I am not into human genetics, but the little I know with the work we have done with mosquitoes is that over the years, man has abandoned, I think that would be the word I would use. Man has abandoned most of the things we did, most of the foods and natural supplements we took in our food that made us stronger and made us to cope with the environment better. And so rather than having a man that is able to withstand a lot of these new diseases and new problems that we face on a daily basis, man had just fallen, in inverted commas, like a pack of cards, because we had abandoned quite a lot of things that we used to do. When we talk about the foods, the vegetables, even the fruits we used to eat and all that, we take more of over-processed foods. And that has actually affected, whether we like it or not, our digestive systems and our metabolism. And so we find that both the various cancers have been something that which we never even used to have in environments here. They are prevalent even from young kids nowadays. So I believe most of the problems we have has been actually a total change of our life, at least in the African setting, a total change of our lifestyles, especially if we believe in the theory that yes, the early man was from Africa, and we know how the early man lived and everything. I'm not saying we should go back to the Stone Ages, but we have within our environment those fruits, those veggies that our forefathers used to eat, which we don't bother about again. We have in the cities, there are people who can live a whole month without taking any veggie at all. And so those are some of the things that have caused most of these problems for us. We can take even COVID as an example. If we look at the prevalence of COVID in our villages compared to the cities, we can find, if we do the statistics, that the numbers are restricted to the major cities. And so is there a form, the question comes, is there a form of resistance that people in the villages still have that we in the cities have lost? Is it due to the fact that we have changed? I don't know. But those are a lot of questions that beg for answers. And it's mainly because, like we said, we don't compile the data when we need to compile the data. And I think it's also as a result of not having a good research funding background. Because if you had a good research funding background with good insurance, even during the peak of the COVID in Nigeria, I'm sure people would have been going around to villages, what is happening, and be collecting this data. But rather, most of the data we want to collect now is after the thing had gone down. And there are those are two various sets of data. But from what we know, most of our villages were not impacted. Most of the figures we got were from the major cities. So has man evolved? Have we changed? Have we mutated? Is there a change in our that is causing that? I would not know, but I would say it is as a result of lifestyle. Our behavioral changes have caused a lot of things to us. And that is why people are beginning to say, let's go back to nature. Let's look at our veggies. Let's look at our plants. Where can we have the antioxidants? Where can we have the immune boosters, which our parents used to eat, that would keep us strong, that will keep us, that will give us a healthy lifestyle and a very good quality of life. Yeah. So that is how I would just answer. I would answer that. Yeah.
Thank you very much, Prof. We appreciate your response to these questions. From the charts, we see that we've got contributions. And if I would like to wrap up this session before we go to the next presentation, it will be to tell us that we have a lot of work to do as a group. What has come up now is that there is need for us to have data repositories. There is need for us to work together as teams to be able to have better research outcomes. If you look at the last two contributions in the chat, it is clear that if we leave all these things to the national bodies, there may be an issue. If we leave it to the government, there may be an issue. But as a group that is concerned about development and health of our environment, we need to work together. Probably we have teams developed. In our group, we have this particular team working together to, address certain focus, and that will also refer us to what the last question we got from Professor Yamaki. He said that this collaboration is key. What can IBS do? What can we do as a group to make sure that we contribute to development? That if you are a statistician, you are a biometrician, you cannot work solely. You need to work with expertise in other disciplines so that we can come up with something concrete, something that is relevant to our national development and even international. For instance, this COVID is an international matter, it's a global issue. What is our contribution as a group that should be equipped with data? If we don't have the data, we cannot actually make an impact. In computer science now, the big issue is big data analytics. What are we doing about it? What are we doing about machine learning? What are we doing about management information systems? It is statistical matter that, these things, they concern statistical issues. And if we do not work with the instrument, other people will work with the instrument, but they won't get the desired result. So as a group, what are we going to do? I believe this meeting should allow us to come up with some positions that will help us. We need a direction, we need a focus, we need a plan. We must have strategies to achieve good results. So I'll ask us to note other issues while we invite the next speaker. We have been able to save about 6 minutes. And Mrs. Oluwashoga, can I ask you to please present the next speaker?
Please, if you don't mind, permit me to exit. I have another meeting slated for 11 with Acting Vice Chancellor, University of Lagos, please.
We appreciate you, Professor Obo.
Thank you very much.
Take your leave. Thank you.
Thank you very much, Professor Obo, for your presentation, and I will be getting in touch with you again. Definitely. Have a good meeting. Okay, thank you, Professor Olaniyi, for the coordination. Professor Obo has opened our eyes to, as we have always emphasized, to multidisciplinary collaborations within the IBS. IBS is not all about statistics. It is not all about statisticians. It is a body of different professions, various fields. Let's come together, work together, and let's move forward, see what we can do to move our society forward through our disciplines. Let me invite at this point Dr. Abidemi Talabi to present his paper. Dr. Talabi will introduce himself better, but from the little I know about him, he's a colleague here at IITA. He has been working, he worked with the maize group, and I believe he's working with the cowpea group now. Dr. Talabi is a breeder and is very, very versed and very competent in his field. Dr. Talabi, good morning and welcome to, he's one of our runaway members. Let me just say that loud and clear. But I believe he's coming back home now. So Dr. Talabi, you're welcome.
Thank you very much, Ma. Good morning, everybody. I don't know if you can hear me.
Yes, we can hear you loud and clear.
Okay, thank you very much.
Yes,
thank you, sir. I would want to appreciate the leadership of this organization for giving me the privilege to actually make my presentation on this platform. Mrs. Oluwashoga has actually introduced me, and I don't think there is really much more to say. I studied plant breeding, and maybe the interesting thing I would want to add is that Professor Fakorede supervised me, and that Professor Mrs. Obo was my external examiner during my PhD defense. So anyway, that's by the way. So now I want to move straight to the presentation of today. I will be presenting on beyond theory, practical application of part analysis to crop improvement. And I know that this presentation actually for many, it will be a refresher presentation, and then maybe there might just be a few new things that some might have the opportunity to see or to relate with. Okay. All right, so I move. So my presentation, I will be having, sorry, please, I need to make some adjustments here. Okay, thank you. So I'll be starting with the introduction to part analysis, and then I'll talk about the conventional approach. I'll talk about the implication for plant breeding, and then I would also go into the sequential part analysis, and I would conclude. I am aware that this society is actually a multidisciplinary society, but I will be talking from the perspective of a plant breeder. I believe people here can actually adapt to their own professions accordingly. One interesting thing about the society is that biometrics brings all of us together, because in whatever research you are trying to conduct, you try to analyze whatever data you have collected, and then you have to be able to present the results to your audience. Okay. So, part analysis. Now, part analysis was actually introduced, or let me say developed by Sewell Wright in 1921. And at that time, the biological scientists were in mind. It was observed that when you are seeing some association between two traits, you might say, okay, this is the correlation between trait A and trait B, but there are so many other interacting traits there that are also making contributions indirectly to the correlation you are seeing between trait A and trait B. So there was really a need to understand what is the real effect of trait A on B, and then what are the other traits that are actually playing out in the model. So that was why this particular analysis was developed in 1921, like I mentioned. Then Dewey and Liu 1959 also explained, and then they defined part analysis, part coefficients rather, as the standardized partial regression coefficients. So the standardized B values actually from your regression analysis gives you the direct effect of a particular trait on another. Now, one good thing about the part analysis is that after your analysis, you actually want to present your results in a diagram. So most times the analysis and the diagram actually goes hand in hand. You don't present a part analysis without a diagram. So as I continue in the presentation, you will see a sample diagram that I want to use to explain what I'm talking about here. All right, so in crop plants, you know, for example, from like I said the other time, talking from the perspective of a plant breeder, we look at yield as our primary trait of selection. But we all know that when you talk about yield, for example, in maize breeding, you also have the yield components. When you're talking about yield components, you might be looking at the hair length, the hair diameter.
Your speaker is off. Your microphone is off. Your microphone is muted.
Doctor, switch on your microphone.
Okay, sorry for that distraction. Okay, can you hear me now? I believe you can hear me now. Okay. All right, so I was trying to say that in crop plants, we have the yield and the yield components, which we can actually look at as a perfect example for the cause and effect relationship that part analysis tries to explain. So talking from this perspective of a maize breeder, like I said the other time, when you are looking at yield as your primary trait, you have the components. For example, you have the hair length, you have the rows per cup or per hair, so to say. You have the number of kernels per roll and all that. So all that actually will give you an indication of what the traits are actually contributing to the final trait of interest, which is the yield. Okay, and then Kang 1994 also explained that the purpose of the part coefficient is actually to help you to dissect the correlation coefficient into direct and indirect effects. Like I said earlier, when you have trait A and a correlation between trait A and trait B, there might be some other traits like trait C, D, and E that are actually having indirect effects. And that is what comes together to show you the correlation you are seeing. So with part analysis, you are able to effectively take out the indirect effects of those other traits. And then you are able to see that, okay, exactly this is what trait A is contributing to the other trait that we are talking about here. Now, when I get to the diagram, I will explain a bit better. Okay, then also I would like to mention, yes, that Duncan 1966 also said that, the conventional regression analysis actually there will be a rationale for it, looking at the part analysis, and basically we use this regression analysis to come up with our part analysis, the part coefficients, and then the diagrams. Okay. So, okay. All right, so somebody might be asking, like, of course, I've mentioned it in passing earlier, that why path analysis? So correlation actually mentions association, but when you want to look at the cause and effect relationships, when you want to effectively know what the contributions of traits are to a particular trait of interest, you start looking at the direction of path analysis. And also, when you are looking at complex traits, there are cumulative influences on a particular complex trait. For example, in breeding, yield is a complex trait for us, and then you have to be able to partition these things you are seeing into direct and indirect effects. Okay, so now with this diagram, I'll just quickly explain what the conventional path analysis does. So for the conventional path analysis, in this particular diagram, it's like we are looking at 5 traits, and we are looking at the effects of 4 traits on the 5th trait, which is the trait 5. So somebody might think that, okay, trait 5 in our case should be yield, fine. But just go along with me and you will get what I'm trying to say. Now, there is a correlation between trait 1 and trait 5. If you look at the first simultaneous equation to your right, now you see that the R15 here is actually talking about the correlation between trait 1 and trait 5. But it would interest you to know that out of this correlation, you have the direct contribution of trait 1 on 5 as P subscript 1 5. Then you also have the indirect contributions. That means the next one you're looking at here, the next component of this equation here, is talking about the correlation between trait 1 and trait 2 multiplied by the direct effect of trait 2 on trait 5. Then it goes along that way. So you will be able to see that in the correlation between trait 1 and 5, you have 3 other indirect contributions from other traits. And then you only have, and then in addition to that, you have the direct contribution of trait 1 on trait 5. And those are the things that come together to give you the correlation between trait 1 and trait 5. So when you run through the correlation, the simultaneous equations that way, you will be able to see that whenever you are looking at any particular trait in relation to trait 5, there are contributions from some other traits, which you have to partition. Okay, and then here also, from the diagram, you see that the P subscript U5 is talking about the unexplained variation. That means sometimes in path analysis, there are some traits probably that you might not have taken into consideration that are making contributions to the expression of the final trait, in this case, trait 5. So that means this is actually going to be the unexplained variable that it's actually coming into this particular model. All right. So what are the implications for breeding? The other time, basically path analysis, like many of you have known, it gives you the idea of traits that are really making contributions to yield. That means their contributions are actually significant, so to say, because so many other contributions might not be significant and it will not be handled in the model. So when you're looking at a complex trait, for example, here, if we're talking about yield, sometimes, some final traits are difficult to handle directly. Sometimes you don't even want to get to the final traits. You want to be able to make selections for the final traits. If you have some other traits that you know make contributions to the final traits, and they are probably easier to handle, easy to measure, and then probably become much earlier and the safety. So in cases like that, path analysis after some experiments have been done gives you an indication of traits that you could actually consider for indirect selection. So in breeding, we call it indirect selection. That means when you are selecting for some particular traits in a bid to actually influence the final outcome of the trait of interest, we call it indirect selection. So path analysis have been used by breeders, and then they construct the selection indices, and then that particular selection index or indices, so to say, are now used for selection for the trait of interest. And here, for example, Badu-Aparaku 2012 et al. Actually use path coefficient and then this GG biplot analysis to identify traits that could actually be used to select, or that could serve as secondary traits for selecting for drought tolerance. And in that particular study, he identified A aspects, plant height, anthesis seeking interval. Okay, now, I mentioned this earlier. Now, I also referred you to Oyekunle and Badu-Aparaku 2013, and there they actually constructed a base index for selecting for drought and low soil nitrogen tolerance in maize. And for that particular index, you would see here the base index is the B high. That means you can have 2 multiplied by the yield. Then when you add it to the years per plant, and then you subtract the anthesis-sinking interval years per plant aspect, and then the staging character, you would have an indication of the drought or low soil nitrogen tolerance level of the particular line you are looking at, or the particular hybrid or variety you are looking at in this case for maize. Okay, so yes, now I would want to quickly talk about the sequential path analysis. Now, the conventional path analysis actually gives you an indication of how the traits are making. Okay, so the conventional path analysis gives you an indication of the traits that are actually making contribution to grain yield, or so to say, the traits of interest. But most of them are just placed as the first order traits. Now, Mamadou et al. 2001 proposed that you could actually categorize all the traits that are contributing to grain yield in orders of their importance, and in orders of their importance rather, and how the contributions are actually felt by the trait of interest. So eventually we discovered that when you have so many traits, if you have just a few traits making direct contributions to your final trait or grain yield in this context, there are also some other traits that are contributing through those first order traits. They are still contributing to what you are seeing in the grain yield. So eventually you can actually order your traits. You can have them in categories of importance. So in this case, this diagram is this is actually from a study conducted between 2011 and 2013. I actually used this for my master's thesis. And then we observed that we have 5 traits in this case that are making direct contributions to grain yield for drought tolerance, or when we evaluated the full seed progenies under drought stress condition. And we observed also that apart from those traits, there are some other traits that are in the second order category, which were also making contributions indirectly to grain yield through the first order traits. Now, lastly, we still have some third order traits that were making contributions through the second and first order traits. Indirectly, they are still affecting the grain yield. So here we are able to see at a glance the different contributions of the traits, and then they were also organized in categories of importance. So, well, somebody might be asking, why do we need a sequential path analysis? It actually helps you to know that no trait is unimportant. You understand? But of course, the category of importance might vary. And that is what sequential path analysis is actually helping us to look at here. So basically, as a plant breeder, when you're actually trying to construct your base index or selection index, so to say, you look more on the first order traits. But now with changing climates and then so many other things coming up that we have not been seen before, we know that we always need to keep revalidating the traits that we have identified earlier as traits that are actually making significant contributions to the final trait, in this case, yield. And also, we know that the second order traits also are thereby considered as potential traits for selection for the particular stress or for the particular interest that we have in mind. So, and then this is what I actually explained here, that the sequential part actually helps you to understand the cause and effect relationship a bit better, because the traits are actually ranked in first, second, third, fourth, and so on order, based on their relative importance in providing explanations to the variation that we're observing in grain yield. And then I also said like I mentioned when we were looking at the diagram, that the first order traits we agree are the major contributors, but the second order traits also are potential candidates for selection for improved grain yield. So I'll conclude by saying that the identification and validation of secondary traits for index or indirect selection remains an important research area in plant breeding. And also the use of the conventional and sequential path analysis will continue to serve as important tools to plant breeders for identification of secondary traits, which can be used for indirect selection. Thank you very much for listening.
Thank you, Dr. Talabi. Can I take over, Mrs. Oluwasegun?
Yes, please, you can go ahead.
All right. Thank you very much, Dr. Talabi, for this insightful presentation. He has spoken to us about the usefulness of sequential path analysis and why it is useful in identification and validation. My observation before I'll let others ask their questions is I'm wondering why you couldn't use hierarchical clustering or factor analysis. I'm wondering if factor analysis wouldn't do what sequential analysis did in your study. I'm just wondering, you may educate me, and we can ask. If anybody has a question, observation, you can please raise your hand now. Please signify. Let's see how many people have questions. Please use the app, please, this is biometry. Don't raise your hand on your video. Just click on the chart to signify. You click on participant, and then on the right-hand corner of the window it will bring to you, click on raise hand, and then we can see you. All right, we have 2 people already. If you have any more, just click on the participant button below the screen, and then it will bring up the participant window. The right-hand corner, you click on raise hand. We will recognize you. All right, thank you. So, Dr. Talabi, let me ask Dr. Oyamaki to give his comments and ask his question. And then we can take your response, and then Dr. Taufiki Dauda will also give his question. Okay, I see Dr. Ladejo. Okay, so Dr. Talabi, you have the floor, please.
Dr. Oyama, can you hear me?
Okay.
For his question.
Oh, sorry, Dr. Oyamaki.
Okay.
Dr. Oyamaki, sorry. Dr. Oyamaki, please, your question.
All right, thank you, ma. I believe you can hear me here.
Yes, please.
You mentioned the first thing I had in mind, and the second question I want to. Well, Dr. Talabi, I love your diagram. I love it. It was cool. It was okay. The presentation was excellent. Thank you, and kudos to that. But I'd like to ask that recently, in some current literature has established some of the superiority of mediation analysis, to path analysis. I wouldn't know what is your take on this. Some people believe that it is, in terms of modeling causal effect, that it is better to go through the use of mediation analysis rather than the path analysis. I don't know what is your take on that. Then secondly, I'd like to ask if we are confronted with some some funny variables, for example, variables that are non-numeric. We have just experienced a pandemic and lockdown everywhere. And let's say we have an example of a variable that exhibits some form of that exhibits nominal features, ordinal features. How do we capture them in this kind of studies? These are just the questions I have. Thank you.
Dr. Talabi, please.
All right. Thank you so much, Sir Professor Oyemaki, for your question. And thank you so much to Professor Olani. You also made similar contribution earlier before the other question. Now, the first thing you asked was that you were wondering why hierarchical clustering or factor analysis couldn't do or wouldn't do the same thing as sequential path analysis. Now, well, basically what I just want to say is that, just like the name mentions, it says sequential. So that means in sequence, For example, if you're carrying out, well, I know, like, first thing I want to identify with is that I'm talking to expert biometricians here, so probably you'll have some better ideas and you could also enlighten us further. But what I want to say is that sequential path analysis, for example, you identify all the first order traits that are making contributions to your primary traits. So you can also regress each of those first order traits on the other traits that have not been accounted for. Then those ones will actually, you discover those that are also making, the significant contributions to your first order traits, and they are categorized as the second order traits. And you can see, of course, each of those ones also have direct effects on the first order traits. Then through that, indirectly, they are influencing the primary trait. The same thing applies to the test traits. It's possible you have all these other diagrams, all these other analysis, probably they are able to solve it this way, but I have not personally used those other approaches. So I think it would be good if we have some people shed more light on that. Probably Professor Imakin, I know, is a professor of biometrics. So he could actually help us with that. Yes, that's one. And then two, Professor Imakin mentioned mediation analysis rather than path analysis. Now, well, what I would say is this. I would also appreciate that it would also shed more light on that. We as plant breeders, most times we use what works for us. When there is an improvement in it and we have seen people using it, we also try to, bring it on board. But so far, so good that I have known, we have been using path analysis now, and the mediation analysis might probably be a better introduction, like you mentioned. So I would just appreciate if you can probably explain how you think that works better, and then we can adopt that. Basically, those are the things I can really mention here.
Right. Thank you. Let's take Dr. Taufik Dauda.
Thank you, Madam. My question has to do with adaptability of this path analysis in other fields other than plant breeding. Because I realized, hello, am I on?
Yes, yeah.
We can hear you.
Yeah, I realized that I was introduced to the package while in Nigeria, but I couldn't lay my hand on it. Unfortunately, on getting outside the country, it was people in econometrics that gave me the package, and they've been using it extensively over there. Though when I examine some of their data fall within the range of using those, some of the features of those package, and it's giving them the result. So I just want to confirm so that at least
Dr. Dauda.
Okay. Dr. Dauda, you may have to repeat your question. We've lost you. And if you're not speaking, please let us all mute our mics, please.
We can move to Dr. Oladejo then. If Dr. Dauda is able to restore his audio, he can ask afterwards. Dr. Oladejo, please.
Okay. Thank you very much for giving me the audience. First of all, I want to appreciate Dr. Talabi Tayo that you just presented. It's a very beautiful and brilliant one. Yes. Actually, what I was just to say, just to make a little comments on the brilliancy of his presentation. Then also to add this, when you are presenting the path analysis, I think path analysis has a kind of advantage over correlation analysis. You remember that it takes care of what you call problem of multicollinearity, and then that of pseudo correlation. So that's just little thing I just want to add to your work. So, and I still believe that in this platform, as we are learning, there is a lot of benefit we are going to share, we are going to get together if we do more of a collaboration. So have a beautiful and a fruitful conference. Thank you very much.
Thank you very much. Thank you very much for your contribution. And I don't know if Dr. Talabi was able to get anything from Dr. Dauda's comments before his audio went off.
Yes, I was able to get.
Okay, you may please respond to these observations.
Yes, what I got from Dr. Dauda's question before he went off was that he was trying to know how we could actually apply the path analysis to other disciplines, other than planning. So what I just want to say is this. Path analysis can be applied to any other discipline. Once whatever traits you are dealing with, whenever you carry out any correlation analysis, then that means you can actually apply path analysis to that particular study that you are looking at. So, but what I want to add to it is that he mentioned package. Of course, we have various packages that have been developed to help with path analysis.
Dr. Talabi, just a minute, please. Just a minute. Dr. Oladejo, can we ask you to mute your mic? Please mute your mic, or let the host mute everybody. Host, please mute everybody, and let Dr. Talabi unmute his mic subsequently. Thank you.
All right, ma. Thank you, ma. So what I was trying to say is that he was asking whether we could apply path analysis to other disciplines apart from plan building, and the answer was yes. Because like I mentioned earlier, it was even the path analysis idea came when they were looking at these biological scientists. So it means that whenever you have any set of traits that are correlated, you can actually partition the correlation, because what sequential path does in this case is that it helps you to isolate spurious correlations from the adjusted values. Now, I appreciate that Dr. Oladejo mentioned this issue of minimizing multicollinearity. Is actually one of the problems that we have using the conventional method, which we have improved on using the sequential path method. So for Dr. Daudu, whenever you have correlations among traits that you are looking at or among variables that have been measured, you can actually partition to know how to know the specific or the direct effect of the individual traits on the trait of interest, so to say. And depending on you mentioned packages, like I agree that there are some packages. We also have some SAS packages that will help you with the normal conventional path analysis. But it's not that difficult. Even if you don't have any package, once you have a package that will help you do regression analysis, then you are good to go. Because from your regression analysis, your standardized b-values are actually the path coefficients. And once you see that they are significant. So that means, for example, if you have about 12 traits and maybe yield is one of those traits, and you regress yield on all of the other 11 traits within that same, then you are able to see which of the standardized b-values is significant. Those are your first order traits. And then, of course, you are testing it's like a t-test. So if you have any package, so to say, once you can run regression analysis within, and you can actually test for the significance of the b-values, then you can actually, so the construction of the diagram is actually manual. Of course, some people also say there are some softwares to actually generate those diagrams automatically, but I believe you can actually do them manually once you have the templates, and that will be fine. I don't know if there is, I think I have.
Thank you. Thank you very much, Dr. Talabi, for the brilliant presentation. There are comments here that I would quickly like to draw our attention to, and it will be for the executive committee, I think, that the slides for the presentation, the members would like to have that. And I checked for Professor Oyemakin, but it doesn't seem like he's online with us. I don't know if there is any statistician in the house who may be able to address some issues that we raised on some of these different analysis that could be done. But I observe a comment also in the chat box from Dr. Akinwale that the relationship between factor analysis and path coefficient analysis is not yet established. I mean, the differences rather in their efficiency is not yet established. I would like to throw a challenge at us. If it's the differences in their efficiency is not established, what are we doing in Nigeria? Can't we have theoretical statisticians and applied statisticians work together to do something and impact, global trend? If this is not done, we are raising these questions. Who can come together and who would like to come together and work? I see that Dr. Talabi has a huge data set that could probably be worked with. If we have theoretical statisticians in the house who will work with you, why not? So that we'll have something published in Nigeria as from Nigerians as our contributions to global development. On this note, I'd like to sincerely thank the executive committee and the organizers for giving me the opportunity to play this role. And I'd like to sincerely say thank you to Mrs. Olayemi Oluwashoga for believing that I could do it, because I had already told her, I said, this is a big work that I cannot do. And she said, no. Thank you so much. I'd like to hand over to you, ma'am.
Thank you very much, Professor Olaniyi. I'm sure you can see that the job is not as difficult as you imagined it to be, and you did excellently well. And I want to thank the presenters too, Professor Obo in absentia, Dr. Talabi. Thank you for your presentations. And to all who have participated one way or the other, I say thank you. Professor Fakurede was supposed to give us a welcome address, but he had challenges at the beginning of the meeting. So I don't know if he has anything to share with the House before we go on to other things on the agenda. Prof, good morning, sir. Professor Fakurede.
Maybe he can unmute his mic.
While we're waiting for Prof to come on, I want to say a very big welcome to our colleagues from Ilorin, Professor Jolayemi. It's been quite some time. You owe me a lot. How is Ilorin? Mrs. Salawu, welcome, good morning. I acknowledge and recognize every member attending. Thank you very much for your time. Dr. Bisheson, good morning. Dr. Folorunsho, good morning and congratulations again on your PhD. Thank you everybody for being on board. Professor Fakurede, if you can hear me, I want to share your thoughts. I want you to please share your thoughts with the House. Okay, while we're waiting for Prof, I sent the meetings of the last meeting that was held at Oka to everyone on the WhatsApp group. I hope we had time to read through. It was a very short, minute. What was not included in the minute were the number of attendants from the meeting. Unfortunately, the person who stood in for us at Oka taking the register did not till now give me the list. But thank God for a medium like this, I can get the list of all attendants at a go. So I'm sure that will not be a problem anymore for this meeting.